Okay, so hi everyone, good afternoon and good morning to those of us in the US. I'm Michael Doron, a postdoc at the Broad Institute at the lab of Juan Caicedo, and I want to share with you a project that we've been working on for the past few months that we're super excited about, and I hope you do too. We are trying to apply self-supervised learning to cellular microscopy and specifically the Human Protein Atlas. And this is still work in progress, but I want to share with you what we've seen so far. The flow of the talk will be as follows. First, I want us to discuss what is self-supervised learning, and specifically Dino, the model that we are using. Then we will introduce ourselves to the task that we are trying to solve, which is the Human Protein Atlas protein localization task. We will explore the feature space learned by Dino on this data, and look at the accuracy of a linear classifier trained on these unsupervised features that Dino learned on the protein localization task. Finally, I want to show you how we can improve Dino and self-supervision in general to work on cellular microscopy images. So first, why do we want to use unsupervised learning in biology to begin with? So we all know the benefits of supervised learning, in which we have the data and we have labels, and we want to learn how to predict those labels from the data that we have. In unsupervised learning, we don't assume that we have any labels. We only have the genes or the images or the time series or the data that we have. So first of all, the obvious benefit is that we can learn features of data without labels, or data with missing metadata, or even combining different data sets that share the same data type, even if not at the same label. A second benefit that I like even more is that using unsupervised learning, we don't bias ourselves to any specific kind of task. If we learn a specific task with supervised learning, we can solve that problem very, very well, but we don't perform as well on other downstream tasks. We can't discover new things. However, if we use unsupervised learning, we don't bias ourselves. We allow ourselves to learn new features, new representations of the data that might allow us to learn something new. In this specific project, we're working with a version of unsupervised learning called self-supervised. In self-supervision, in this case, something similar to contrastive learning. What the model does is that if this, on the upper left corner is the feature space, the model takes these two samples, or images in this case, and creates many different versions of them, many different augmentations or views, if you will. So you see this one image here, and it has many different versions, all of the same image, unlike this version, which is of a different image. What the model does in this self-supervised learning is that it tries to learn a similar representation to all of the same versions, all of the versions of the same image, while learning a different representation further away in the feature space for versions of different images. This allows us to actually learn a compressed version of the images of samples without having any labels of them, only the data itself. We will use a specific model published recently by Facebook, which is called Dino, for self-distillation with no labels. And the interesting thing about this model, apart from the fact that it uses self-supervision, is that it uses vision transformers instead of convolutional neural networks. This allows us to extract better features when we have large amounts of data, which is what usually happens in experimental data and in unsupervised learning. So our question was, can we use this self-supervised model, this Dino, on biological data? To test this, we took the Human Protein Atlas protein localization task dataset. In this task, we have human cellular cultures of different, and these cultures were treated with antibodies that bind to different proteins of interest. These proteins can localize in different areas inside the cell, and our task is, given those images of the cells of the cultures, our task is to find where do these proteins of interest localize, whether it is in the nucleoplasm or the plasma membrane or the cell junctions, or in this case, all of them. This problem is particularly difficult because in almost all of the data these proteins localize in more than one region in the cell. In addition, this problem has a huge class imbalance where more than half of the images have proteins that only localize in one of two classes. The rest of the data has proteins that are localized in the other 26 different regions in the cell. Meaning that this is a very difficult problem to solve, and we try to see whether we can solve it without using any labels. So we applied the Dino, we trained this self-supervised model on this data. What you see here is the UMAP embedding of the feature space learned by Dino on this data. And you can see that without any labels, we already see some interesting structure here. We already see some isolated clusters. So we thought, okay, we may have been able to solve it from the get-go. But when we color these different samples by their protein localization class, we don't see any particular design or structure here. In fact, it seems that every one or almost every one of these clusters exhibits all the different protein localization classes. So this was unfortunate, but when instead we color each one of these samples by their cell lines, we see that this is what the model learned. So in other words, without using any labels, Dino was able to find the biggest factor of variation, which is the cell line, and the second factor of variation, which is the protein localization class. After that, we trained a linear classifier on top of these features, meaning no new feature learning was done. We took these features and we trained a linear classifier on top of them to solve the cell line prediction and reach a very good result. Meaning that, yes, we can use Dino on biological data and get meaningful features. But how meaningful are they? When we try to solve the more difficult task of the protein localization task, we didn't perform as well. What you see here is the F1 score of all the groups from Kaggle who tried to solve this problem in a Kaggle competition from 2018. And you see that Dino reached the 1660 place among all the other groups that tried to solve it. This may be impressive considering that we didn't use any labels in the training and all the other groups used supervised learning, but still we wondered, can we do even better? So in order to test this, we chose a smaller data set from the bigger human protein atlas data set that only have 5 different protein localization classes, 5 instead of the 28. We used 2 baselines. One is the second best performing group on Kaggle that tried to solve this task, and the other is a convolutional neural network that was pre-trained on another dataset on ImageNet. And we tried to see which features allow the best performance. And we saw that although vanilla Dino without any changes does not perform as good as the groups that try to solve this particular task, we still perform much better than the other pre-trained networks. So now we will try to see whether we can improve it even more. You remember that earlier we discussed the way that self-supervision works with the different views or augmentations of the different images. These are the augmentations that Dino came with, either flipping the image, cropping and resizing it, solarizing the color, blurring, jittering the color, or grayscaling. And we thought that these all might not fit cellular microscopy. So we thought of a few more: rotating, removing the channel, mixing up different patches, or rescaling the different channels. And we try to add or remove each one of these augmentations to see how well we do on the task. And we saw that some of them, like grayscale, should be discarded completely. Some, like rescaling the protein channel, should be added. And we took all of the good ones, removed all of the bad ones from our model. Next, and this I think is the most exciting part, we try to see whether we can improve Dino by taking these full field of view images and dividing them into single cells. So we use a standard segmentation method. We use the cell pose, and we took these particular cells, and instead of having only these full images, we multiplied our data about 20-fold, because there are about 20 cells on average in each cell, in each image, sorry. And this allowed Dino to reach better accuracy and have better variability, since now we're looking at each specific cell, not at the aggregate of the entire image. There were other kinds of improvements that we had that I don't really have time to go into, but I will mention just briefly. We reduced the patch size that Dino looks at, meaning we increased the resolution. We added parameters to the model, to the vision transformer, trained for longer, obviously. And an interesting part, we trained several models, one on each individual channel, in addition to one on the entire of 4 channels, and concatenated the features, the feature vectors learned by this model, reaching much better performance. And then once we look at the performance of all of these changes, we see that while we don't yet reach the Kaggle performance of the people who use supervised learning, we still improved our performance dramatically compared to where we started. And this is just the beginning. We did not yet try to combine all of them, or whether we did try, it just takes very long to train. So this is training as we speak, and combining all of these different improvements, and who knows where we will reach once we combine all of this. And in the short time that I have left, if I have, I want to show you a bit about how we can use Dino for discovery. And what we have here are our 2 cutoffs of the cells from the same culture. So you see that this cell was supposed to be here, this cell was supposed to be here, etc. And what you see here, that on the left are all the cells that received correct predictions using our classifier on the Dino features. And all of these cells received incorrect predictions for each specific cell compared to when tested on the class of the entire culture. And what I think you can see is that visually there seems to be some difference between these incorrect prediction cells and these correct prediction cells. And what we think is that it could be that some cells just didn't have the protein localized in the same place as the entire group, as the entire culture, and Dino was able to catch that because it learned those features in an unsupervised way, and we didn't force them to be features where this cell shows the same protein localization as this cell. And finally, when we look at the embedding themselves, that again we learned in an unsupervised way, we see that in this cluster right here, there are 2 types of cell lines. There's the CH line and the HeLa line, both of which are cancerous epithelial cells from the female reproductive system. So what we see that without knowing anything about these cells, Dino was able to learn the correct features that put similar cell lines in the same place. And this is all work in progress, and we hope to show even more exciting things later on. And to conclude, vanilla Dino cannot yet, and maybe shouldn't, replace supervised learning, but it can extract meaningful features from biological data in an unsupervised way, and these features can be used to solve both the cell line, which is the easier prediction task, but also partially solve the harder protein localization task. We can fit Dino to solve cellular microscopy tasks by changing, adding, or removing the augmentations or views the Dino uses, training on single cells instead of full cultures, increasing the vision transformer resolution and parameters, and finally combining models that train on different aspects of the data. We already showed some meaningful semantic meaning in the features, and hopefully we can use more. And that's about it. I want to thank my colleague Wolfgang Pernis from Columbia University and my advisor Juan Caicedo from the Broad Institute, and thank you for your attention.